Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So today we will discuss about the microbiota in our gut and production of useful compounds. So the useful compound which that is undigestible in our gut is digested by the bacteria, which that is known as the microbiome, and that's microbacteria, basically bacterium which that is used for it. So here is a person which that is containing a carbohydrate and some food, which that is carbohydrate is made up of amylopectin and amylose. On the other hand, the homopolysaccharide or heteropolysaccharides containing compound. Homo means same glucose molecule combination. On the other hand, hetero means the different combination of the glucose molecule. So when that's go into the inside of the stomach, that's digestion of carbohydrate will be start from the oral cavity and go into the duodenum, and further digestion will be occur, and then go into the large intestine, and after the large intestine all absorption will be occur of the carbohydrate, but the remnant nutrient which that are present in the large intestine is absorbed by the helping of the bacteria. So here is the symbiosis or mutualism is known. That's a type of symbiosis mutualism, which that is or commensalism, that bacteria provides short chain fatty acids to the body by fermentation. So the what type of short chain fatty acid, which that is less than 10 carbon containing fatty acid. So here is the large intestine. So here we will zooming the large intestine to understand the function of large intestines, and their inside is a layer is the epithelial cell, which that is known as the enterocyte like structure. So here is the ascending colon. Ascending mean which that is going upward, and that's come from the small intestine, when small intestine attached from here, and that's the transverse colon, and transverse colon give the descending colon is the downward. So here is the sigmoid colon, which that is a sigma shape. On the other hand, here is the rectum, which that is basically rectum is used for the storing of the waste material. On the other hand, here is the anus, which that is allowed to move outside the feces, is known as the sphincter muscle is also. So one by one we will discuss about the large intestine's layer inside as well as the small intestine, and so the difference of that we will discuss here. So on the other hand, here is the microbiota, accessible carbohydrates. So the accessible carbohydrate which that is come into the microbiome, and that means the large intestine is not able to digest, or our gut will not able to digest the fructooligosaccharides and cellulose. On the other hand, the resistant starch. So the cellulose is the type of polysaccharide which that is the present in the plant wood, so that is used for the digestion of the ruminant animal, for example the cow and goat. On the other hand, here is the epithelial cells are present in the enterocyte, and that is the epithelial cells upper layer is not a brush border like structure, and that is used for the absorption of water as well as the some nutrients, some ions. So here is a layer is known as the mucosal membrane or the mucous membrane. That layer is the very much a barrier for the invading of the microbial invaders invading inside. So the resistance will be created by the mucus barrier. So that's here is the microbiota, and here we will discuss about the small intestine. If we will talk about the small intestine, which that is here is the esophagus and stomach, and that's the duodenum. So the duodenum contain a less amount of mucous membrane. Why? Because it's acidic medium, and this acidic medium will prevent the infection of the bacterium, which that is the unuseful bacteria. So the unuseful bacteria is not infected due to the acidic medium. On the other hand, that's less amount of bacteria are present, and that if the bacteria will come into the small intestine, that's the acidic medium will be neutralize the bacterial enzyme. So that is why the layers is the thin as compared to the large intestine. So I hope you make sense about the large intestine. That's a colon. Is the basically the cecum contain a large amount of the bacteria. So on the other hand, that is the harmful bacteria which that is not infected to the large intestine due to the large amount of mucus layer. So here is the bacteria type. So the primary degrader is known as the bifidobacterium species, as well as bacterioid species, and Ruminococcus bromi, which that is ruminant animal also contain it, and these digest the cellulose. So the produce short chain fatty acids known as the propionate, and glucose is the one of the most example, as well as the glucose is come from the cellulose and resistant starch. On the other hand, the fructooligosaccharides. So here we will discuss about the microbiota accessibility of carbohydrates. For example, the accessible carbohydrate, which that is the undigestible in our gut, which that go into the large intestine, and this type is the here, which there is the fructooligosaccharides, cellulose, and resistant starch. So the fructooligosaccharides structure here is the basically resistant starch, which that's resistant starch mean the the number of sugars is attached by the alpha and beta unit of glycosidic bond, alpha and beta, and that's complex molecule is not digestible because the lack of enzyme are present in our body, which that is the lack of their digested enzyme. And cellulase obviously are not present, which that is only present in the ruminant animal gut. So that cellulase is not present, and here is a fructooligosaccharides, which that is the resistant starch or resistant polysaccharide, as basically oligosaccharides, and these oligosaccharides contain fructose as well as glucose, and that's the alpha and beta glycosidic bond are also present. Anyhow, here we will discuss about the primary degraders. So the primary degraders is, first of all, we will understand and production of the short chain fatty acid from primary degraders. So the primary degraders is the bifidobacterium species and bacterioid species, as well as ruminococcus species, which produce the short chain fatty acid as well as glucose. But here is the structure we will discuss here. That's the bifidobacterium species is the largest as compared to the bacterioids. So the bacterioids is the smaller part of the bacteria, is basically the small bacteria, and these bacteria is look like this. And coccus mean that is the cocci shape, and rumino mean the ruminant animal, which that contain these bacteria is also. But human gut contain less amount of this, and which that digest the cellulose. So here is the acetate, basically the propionate produced, that is a short chain fatty acid, and here is the formate, which that is also the short chain fatty acid, which that is the less than 2 carbon. So here is the glucose and acetate that's produced, and this short chain fatty acid is useful for the human gut, and this short chain fatty acid is go into the liver to further digest and produce cholesterol or use as a energy. This short chain fatty acid is very important for our health, and which that is depend on the basis of basically that depend on the basis of age and exercise or some other environmental factor. So here is glucose is allowed to add the secondary degraders is known as the firmicutes species, and that's firmicutes species is digest the sugar, and that's convert into the butyrate, and this butyrate is the short chain fatty acid. On the other hand, the acetate is also produced from the primary degraders, but here is the 3 carbon containing butyrate will be produced. Butyrate. So here is the cross feeding is also, which that here is the acetogen, which allow to eat the formate to produce butyrate, and that is basically butyrate produce or produce a acetate again from the formate. So this is the cross feeding, which that is the primary degrader is also produced acetate, but formate is also absorbed from the cross feeding bacteria acetogen to produce it acetate. So on the other hand, the methanogens is meaning the methanogens methane producing bacteria which produce the methane gas, and this gas is the character of the fart during gas production from the during fart, that is due to the methane gas production. So on the other hand, here we will discuss the important notes which that waste food can be helpful by the bacteria such as mutualistic activity, and that's bacteria uses glucose for glycolysis and provides short chain fatty acids as a byproduct for human health. So these bacteria is changes by the age, exercise, genetics, and environment. So on the other hand, important to know that the bacteria which that is anaerobic environment creation. So the anaerobic bacteria is allowed to enter that things. So that bacteria is not aerobic. So, a large amount of anaerobic bacteria because a less amount of oxygen are present in the large intestine. So that is why it's consume sugar to produce acetate, and consume sugar to produce that is known as the starch fermentation of the bacteria in the large intestine. So remember about that. So here I am drawing that is the hepatic propionate metabolism, that's a short chain fatty acid, which here is the large intestine, same here, and that's contain enterocytes or epithelial cell which contain a mucus. So the all sugar, acetate, propionate, and methane, which that is the methane and propionate, which that is enter into the bloodstream, but methane will not enter. Methane is going into the fat, and that's released as a gas from the digestive system. On the other hand, that's a propionate. That's propionate which that is enter into the enterocyte directly because it's a diffusion. So it is also attached with the LDL chylomicron, and chylomicron will attach to carry and transport into the liver. So on the other hand, here is the acetate will also move inside. On the other hand, that is the sugar which that is allowed to move inside by the active transport by two sodium ion move inside, and here is the potassium sodium ion channel exchanges to move sugar inside. So here the acetate and propionate will be enter inside, and that's go into the liver. When go into the liver which that is directly receive that's a propionate, and this propionate will be convert, here is a propionate, and which that's convert by the coenzyme addition in the presence of ATP to convert AMP, and that's 2-phosphate used for it to make a propionyl CoA, and this propionyl CoA is use the biotin and magnesium ion to as a cofactor to produce a methyl malonyl CoA, and this methyl malonyl CoA will use the coenzyme also, coenzyme B12 to produce succinyl CoA, and this succinyl CoA will go in the, that is the character of tricarboxylic acid cycle, which that is a Krebs cycle to produce ATP as well as carbon dioxide. So that is the mechanism for it, and that's used easily as compared to the long chain fatty acid. So the short chain fatty acid is healthy as compared to the long chain fatty acid because it's metabolized very quick and fast easily. So here is the acetate. Basically here is the acetate, which that is the reactive oxygen species containing acetate. So this reactive oxygen species is the harmful for the tissue and DNA damage, and there are many damage. So that's convert by the ACSS, convert by the acetyl CoA, which that is the acetylation to the another molecule, as well as lipogenesis will be occur and go into the adipose tissue. So the acetate reactive oxygen species will be released to convert again that things. So thanks for watching.